Alright, welcome back to class. We will be looking at planning an investigation today. How do we plan a biological investigation? That's what we'll be looking at. We'll be looking at the steps involved. We are going to work with a mnemonic that will make us remember the steps. And we are also going to look at a sample of questions to test our knowledge, to check for our understanding. So let's go as we start with the mnemonic. So, I don't care, so run away. This, I believe, is a very simple mnemonic that we can easily remember. So let's look at the meaning of each of these. I goes for independent variable, which is the variable you change in an experiment. This variable usually has to be stated. You have to state the name of the variable you change. You have to give it a value, that's the quantity, and then don't forget its unit. If you have to use a particular instrument to determine this quantity, please state it clearly here. Then a dependent variable. This is the variable you measure. You have to also state the measuring instrument you use. Again, don't forget the unit. Later, we'll get the clues on how to identify them in experiments. We have all other variables apart from independent and dependent variables referred to as controlled variables. So these variables must be kept constant, and they may be up to 2 or 3 and can earn you 2 to 3 marks in planning an investigation. Now we have safety. You need to look at the potential risks that are surrounding the experiment, and you may also be required to give a possible way out, to find a way to eliminate those risks. So then we can talk about repetition. Any experiment that is deemed valid has to be repeated, especially to remove anomalous results, and then for it to be reliable. Then the last, taking average. Having gotten 2 to 3 or more repeated values, it is essential you take an average to get more accurate results. So remember the mnemonic again: I don't care, so run away. So the reason why planning an investigation becomes a big deal in IGCSE biology alternative to practical is because it carries the most mark in the past papers. So it will usually take 6 marks. So I believe nobody would like to toy with 6 marks. So let's look at a sample question that has to do with amylase. This is an enzyme that we know breaks down starch into reducing sugar like glucose. So the question says, plan an investigation to determine the effect of temperature on the rate of breakdown of starch by amylase. Now, from the title of an experiment, you can easily determine the independent as well as the dependent variable. They are usually well represented in the title. The first factor they usually mention in the title is independent variable, and the second one is dependent variable. Sometimes it may not come in that order, but you just need to look at the title to find out which one depends on the other, or which one will be changed such that it can affect the other variable. Alright. In this example, of course, we can easily identify temperature as a dependent variable, so you need to talk about how to change this temperature. So you can talk about 2 or more different temperatures, that's about changing it, and then you will need to state the exact value you want to work with. Are you looking at 20 degrees Celsius and then maybe 40 degrees Celsius if you are going for 2, or you can go for even 3 values, but remember to put their units. Remember to state the instrument you will use to arrive at those temperatures, and remember to state the exact number, the quantity. So the variable we are measuring, since we are dealing with rate of breakdown of starch, anything like rate in science or biology usually has to do with time. So now we are looking at how the starch is broken down over time. Okay. So of course the use of stopwatch and stop clock will be necessary here. So the method you use to arrive at any of these variables, or how they are measured or determined, need to be well stated. Now, if you look at the dependent variable, we talk about the time taken for a positive result for reducing sugar, or time taken for a positive result for starch, for negative result of starch. Why do we have to go in these two possible ways? Very easy. Amylase is an enzyme that acts on starch. When it does, starch becomes the substrate of this enzyme, and the product, of course, is reducing sugar. So in a reaction, in a chemical reaction like this, you consider the time, the rate at which the reactant, the substrate, disappears, or the rate at which the product, this time the reducing sugar, is formed. So we should expect that the starch will be reducing here until it's no longer present, when all has been catalyzed by the amylase. Okay, so it will start by turning blue-black, because at the beginning starch is still abundantly present, until the reaction is complete, when we no longer have starch. That time we have a negative result for starch, and we know that the solution will be orange in yellow, or yellowish orange, which is the color of iodine. Okay, initially it will be blue, turning blue-black, because at that point the starch is still present in excess. So if you are going in the way of starch, you do that. But if you are going in the way of glucose, initially there will be 0 glucose. So you keep testing for glucose, and there will be more and more and more glucose present until it turns brick red, which is a positive test for glucose using Benedict solution. So it's also important you state the details of the method you use. You are measuring glucose with what? With Benedict solution. You are measuring starch with what? With iodine. So you need to specify, you need to also talk about the time intervals where the measurement will be taking place. Is it every 5 minutes? Every 10 minutes? So you need to state this very clearly. Or is it every 2 minutes? Okay? You have to state this very clearly. Those are the details of the method that you need to represent. Then all other variables, remember, has to be kept constant. And when we are talking about constant variables, there are some you can easily pick out. For example, here we are talking about enzymes. We have known before that pH affects enzyme activities. We have known before that temperature affects enzyme activities. Now that we are dealing with temperature in this investigation, you should remember that pH has to be kept constant. Okay. Another thing you can think about is, if you are working with time, like in the case of determining the rate, the time interval has to be the same. If you are using 2 minutes space, you have to use that throughout. And then there is one other part that is always almost present as controlled variables: concentrations and volume. So if you are talking about any reagent at all, the concentration of that reagent is a mark on its own, and the volume of that reagent, which has to be the same, is another mark on its own. Like in this case, concentration of starch must be kept constant, the volume of starch must be kept constant, concentration of amylase must be kept constant, even with the testing reagents, the Benedict solution and the iodine. If you are using drops of iodine, it has to be the same. So please, let's take note of this while we proceed to the next one, which is repeating the experiment and then determining the average. In some past papers, these two will be seen as a singular mark. You repeat the experiment. Having gotten these extra values of the repeated experiment, you need to take an average, and finally safety precautions. So you need to look at areas of danger, aspects that that one need to take precaution about, okay, in the entire experiment. Like if you are working with Verdi solution, there will be a point where you have to hit. So it will be better to work with water bath than working with Bunsen burner to arrive at this. Okay, now is a classwork for you. Can you try to apply the knowledge you've learned so far to solve this question? At this point, you may decide to pause the video for some minutes to get this done, maybe 5 minutes to answer this question. Okay, so now that you are done, can you try to look at the masking? Are you correct? Did you get it correctly? Mark it for yourself and learn from there. Thanks so much for taking your time to follow the class. Help us to like, help us to share, and subscribe to this channel so you can get notified anytime new content are updated. Remember to share with friends, let them come and learn and enjoy the beauty of biology. Thank you so much for your time. And in the next class, when we meet again, we'll be going to part 3, which will be food test. See you in the next class. Have a nice day.